My name's David Finkelstein. I'm a professor at University of Melbourne, Australia, and it is my great honour to interview Professor George Perry for the Journal of Aging and Neurodegeneration. Professor Perry holds a Semmes Foundation Distinguished University Chair in Neurobiology at Texas. Dr. Perry is one of the most highly cited Alzheimer's researchers in the field. You have been the chief editor for Journal of Alzheimer's Disease, fellow of the American Society. You've received numerous honours, including the Denham Harmon Lifetime Achievement Award, the American Association of Aging, and from the Zenith Awards from the Alzheimer's Association. You're a member of the Spanish Royal Academy Society, a foreign member of the Mexican National Academy of Sciences, and you take great pride in now Azorean-Portuguese heritage. Is that correct?
Correct.
Yeah, and you've served on the advisory council. You have so many honours and awards that it's too many to read out for. But I think your work has been centred around Alzheimer's, oxygen radicals, oxidative stress, and the causes of Alzheimer's disease. Some questions. What initially sparked your interest many years ago in Alzheimer's disease and neurodegeneration?
Employment. Totally. Explain. Employment. I'm trained as a marine biologist, and I did oxidative stress work in marine biology, and I did a postdoc in something else to complement that in medical research at Baylor College of Medicine. And when I finished that, the employment options between medical research versus marine biology, could say they were 100 to 1, but it probably was 1,000 to 1. And so therefore I ended up getting recruited by Pierluigi Gambetti for a large program grant to do research on Alzheimer's disease.
So when did it grab you? When did it become a passion? Because clearly Alzheimer's is a passion for you.
Not for quite a while. I think it became more of a passion when I obtained tenure. So that would have been in the late 80s. So 6 or 7 years after. When I realized I was probably going to succeed in Alzheimer's disease. And further, that I had to figure out a way to do something in Alzheimer's disease that was similar to what I did as a marine biologist.
What was that?
Oxidative stress.
Yeah.
So I invested 7 years to get the first paper. So I invested probably 20% of my effort to figure out what I could find about oxidative stress and Alzheimer's disease that would be at least partially credible. And that was not trivial at the time, about what I could measure, about what I could demonstrate. And with that I became much more interested in Alzheimer's disease.
That's fantastic. I wish people could follow in those footsteps and follow and derive quality research, work out where they're going and their passions. That would be wonderful.
So I didn't, at least initially, I had very few footsteps to follow. But even my work in marine biology and oxidative stress was pretty original. I studied one phenomena that was a naphthoquinone oxidation that the prior papers were published in the early 1900s, and another piece of lipid peroxidation that was also pretty novel. I mean, I was copying work that was published in human blood in sea urchins, but the metabolism was quite different.
Do you think that's missing, that quest for originality now? Because there seems to be a lot of follow the leader, follow the theme.
I have that when I have people working with me that constantly show me papers. He won't watch this video, so that'll be okay. They'll show me papers that other people published, and they said, this is where the field's going. And I said, I really don't give a blank. Yeah. I really don't care what other people do. I mean, I need to know what it is, but I don't bother to do what they do. When I write papers criticizing other people, some people will say, why did you do that? And I say, because it makes me happy. It's in the part of my expression.
It's happiness in science is very important, and you have to lay in bed and you have to be able to do the best science you can. Yes. Clearly you are and have.
Well, thank you. You're saying that. I basically try to figure out how to solve problems. Mm-hmm. And I don't know about if I know how to do them in the most sophisticated ways. I figure out how to do them. They answer the question the best I can, because science is a process. And it's important to know it's a process that, and by that I mean that there is no definitive answer, usually. I mean, my work that I published, the work I did in my thesis, it's still unclear what it is. I think I did the naphthoquinone project I did, which was the second project I did as a student, is probably the best study I ever did. It's the most complete. I started with activity and I came out with crystals and I worked out the stoichiometry. Do I know it's important? I have no idea, but it's still a really fulfilling experiment because I know it's definitive. What I demonstrate is definitive. What it means, I don't know. And I think that's true for a lot of things. The best you can do is be able to show reproducibility, quality. Instead, I think there's all of this stuff of people following what other people tell them is important. So do I do that sometimes? Sometimes, but most of the time not. I'm not I don't really get self-reinforced by people telling me how smart I am or any of those type of things. I mean, I don't know. I'm not that swift, but I know I can be really persistent and I'm not stupid. So I think I can solve things differently than other people. And I think a lot of people are trying to become famous. And I'm not against becoming famous or any of those things. I'm not against publishing in top journals. I'm not against any of them. But that isn't the main thing. The main thing is finding new things. And if you follow other people, your chance of finding something new is almost zero.
That's fantastic. Thank you. Could you share some of what are your current research projects and what drives them?
What drives it for me? Yeah. I made a list and I have to find it. I made a list of 5 things I was interested in knowing before I finished my career. Am I pursuing those? Tangentially. I want to understand the nature of how oxidative stress occurs in real cells in real living systems, not just cultures. And to do that, I've been working on, and I want to understand what the role is of amyloid and tau in the disease. I think they're compensatory mechanisms to the disease.
Ansynuclein.
Oh, all of them. All of them. All of these filaments, all of these proteins. I think that what we found by studying them really are the key elements of the aging nervous system. That these proteins, when they get overproduced and they form fibers eventually, like amyloids do, but they have a biological purpose. And the simple act of removing them is really, it's like beyond stupidity of biology. Responses that are common in well-adapted organisms are not going to be detrimental. That doesn't mean that they can't be detrimental, but that isn't the way in which people have said it. They've made it always seem like they're evil, which comes back, to medieval exorcism and bloodletting and all sorts of things that physicians do. So I've been trying to understand the exact mechanism responsible for this. And I thought it would be really easy to figure this out in plaques. And I have explanations, but I'm not happy with the explanations that I published earlier. Not that they're wrong, but I don't think they're the right explanation. They're not incomplete. And that is that amyloid beta binds copper, which is a redox active metal and important in catalyzing oxidative stress, and it changes it, blocks it. The reason I didn't like that is it's stoichiometric. And stoichiometric processes probably are not big time important in biology, okay? Can be. So I kept thinking that we had published even earlier that the plaques and tangles had ferroxidase activity. We didn't call it that. We published it. I don't even know if we knew to think of that. But anyway, we call it iron-related redox activity. So I really wanted to understand what is the redox activity, what is the state of iron in plaques and tangles. So I thought I would do plaques first because I'd wrap it up in like 6 months. Well, I've been studying it now for years because they're easy to isolate, but the state of the oxidation of the metal is really complicated. So what we found was the iron thing. We found that we could find iron 2 and iron 3 crystals. Some of it's in the form of magnetite. And eventually, working with Joanna Collingwood in the UK, we were able to determine the redox state of copper. And it's a copper 1 and copper 2, which is not trivial to identify using the X-ray synchrotron-based microscope. But the thing we found more recently, and so bizarrely, is the dominant form of the metals is metallic. Zero valence. Zero valence has never been described in biology.
So lumps
for the case of iron. Not lumps
of iron?
No, it's the lumps are magnetite. There's magnetite crystals in plaques that are very small, but they still have some magnetic properties, paramagnetic properties.
Okay.
And that we determined by a few different methods.
Yes.
Which is not trivial, that whether it is magnetic or not. Although we knew it was magnetic for years. But even that, our determinant is Mark Smith, my close collaborator that died over a decade ago. He found that when you isolate plaques and tangles, they would move around on a magnet. You could just take a magnet and move them around.
Wow.
It took us how many years? 20 years to figure out how to write something that didn't have a picture of my hand with a magnet. Okay. And so it's probably both the iron and iron oxides and the copper. And it's probably present mostly as some kind of thin film on filaments. The copper is present. I think that I also wanted to know the copper binding sites. I think I virtually killed 2 or 3 postdocs with this thing because we still never know. We do know it from an earlier experiment, but that was very crude. That was Raman spectroscopy. But trying to do it in a more detailed way where it binds, it doesn't look like it's binding to the histones. I mean, the histidines, excuse me. The histidines that are present, the obvious sites. In vitro, yes. But in vivo, actually some experiments we have, which still are not as good as we would like, they are suggest that the copper plays a role in binding together the filaments. So all of this, what does it mean? There's a lot more chemistry. So the last paper we published together with Collingwood et al. Is that we define the same results if we don't do anything to the tissue. So it's not a matter of, running through isolation procedures for the plaques. Even if you take tissue and don't do anything to it and look at it by x-rays, and they develop some techniques to be able to look at things. And then they've also been doing work to see if you could produce iron metal under conditions that would be biological. So iron metal does not exist in the earth almost at all. The main source of iron metal is meteorites. There are there's some rare ores. There's some rare places on Earth because it rusts.
Yeah, we've got the whole. If you fly over Australia, the Western Australia,
that's big.
It's a huge area where it's just red because of the abundance of iron oxide.
Exactly.
Can I take you back one step? When do you think Alzheimer's begins? Because you've suggested that
Not this
copper,
is I don't think okay. Let me tell you.
Oxidative stress. Yeah, go for it.
Yeah, where do I think it begins? I think if I were having to guess based on weak data, I would say it's a metabolic disease leading to an inflammatory disease, leading to neuronal failure in various ways. So other experiments, there's a very interesting paper that was published in PNAS this month. I wrote a commentary for it, in which they induced in mice with normal genes, but modifying the immune response genetically, and they got plaques and tangles. Remember that mice are not supposed to be capable of generating plaques and tangles. They were as good at tangles as anybody manages to generate, but they generated real plaques and they had behavioral problems. So my interpretation of that is that inflammation is above many of these other responses. The oxidative stress, I think, is a little bit of a later event. And I think that it relates more to how mitochondria are turned over in cells. So we've documented in the past that mitochondrial issues are an important element, that mitochondrial garbage accumulates in people after the age of 40. By apoptosis rules, the cells should all be dead, right? When you release mitochondrial proteins, things are supposed to go south. Well, they don't.
No, they don't.
So you end up with a small amount of normal people over 40 and much more when people have Alzheimer's disease. And what mitochondria are bags of metalloenzymes. All those oxidative enzymes are copper and iron enzymes. And if you're turning over, we documented the DNA portion, and we also saw some things with proteins. I suspect what's happening, if I were guessing, and I actually haven't written this in a very clear way, is that mitochondria turnover changes in some way. So the end out, you accumulate a lot of mitochondrial garbage, and eventually, normally it would have gone to lipofuscin. Yes. And the lipofuscin in Alzheimer's disease is very different than the lipofuscin in normal people. I haven't published that.
I didn't know that either.
Well, it's not more, although people have published that. There's slight changes, okay? Slight things. But one of the things is it also has this property of heavy metals. So it isn't that there's more metal. It isn't that there's more metals, particularly. I know Ashley Bush says that there's more, and there could be. But I know meta-analysis that people have done saying that there isn't any difference. Our question with the metals isn't about them being more. It's about their redox state, about how they're accumulating. Now, finding them with the metals. So if you isolate, when you prepare plaques and use sucrose gradients, the very bottom of the gradient's lipofuscin for Alzheimer's disease people. It isn't for normal people. In fact, I don't even know how to isolate them from normal people. So there's a big change, and it has to do with probably mitochondrial garbage. Now, does that deal with the issue of it being primary? I don't think so. The other part, I will tell you, and this is a paper I've had the data for 2 decades and have not put it together. I promised myself, I tried to suck the energy out of other people to help me on it, and they haven't been able to. So one of the things is I actually don't think the disease begins before. I think changes begin before. So the idea that
prodromal.
There's prodromal because we're prodromal for everything.
Yes, that's true. But
we're prodromal for death. We're pro. In fact,
No.
I can offer diagnostic services for lots of different things for very low cost, $10 per person, $25 per family, and I can diagnose pro death.
Yes.
So what does it mean if you say that you have a disease 25 or 30 years beforehand, unless you have autosomal dominant disease?
No. I think what they're saying now is that there are symptoms which aren't picked up clinically that you can pick up in the lab.
Oh,
that's that's my definition of prodromal.
Yeah, I know. But, a lot of this is new ways to scare people so they end up taking a bunch of risky treatments. Or donate more money to research or do something else.
Now, post-polio syndrome was when I started my PhD many decades ago, and when people recovered from polio, then all of a sudden as they age, they dropped out, their neurons dropped out. And I think this is an analogy to what you're saying. We know that we lose neurons from the age of 20. If you look at those histological studies,
yes.
At one point, that's normal aging.
We know as we become age, we're going to decline and die.
Yeah,
that's predictable. So Alzheimer's disease, does that negate that we can have biomarkers that can change your risk? Okay, yeah. I think it's different for other people. A lot of people are motivated, especially young people. They think if they get a paper in Nature that their life is, easy street, that they're close to God and everything else. Yeah, I could have that feeling. It might last, 25 minutes. But that's more than it normally would. Let's say I get excited by a paper 10 minutes or something. The challenge in this particular case is that so I looked at the relationship of mitochondria, oxidative stress, and amyloid. So all my publications on this topic, I found the inverse relationship. The more amyloid there is, the less oxidative damage. And that's one of the bases, not the only one. Base I came up with the idea that maybe amyloid is protective. But I will tell you, in this study, which included normal people, which none of the other ones really did. They were there, but they weren't analyzed. Normal people actually have a very different situation. Their oxidative damage is much lower. It's related to the amount of mitochondrial DNA so a mitochondrial garbage in cells with an R squared of 0.9.
Whoa.
So highly correlated. The inverse relationship for amyloid is also over 0.9, 0.95s. But for normal people, the amyloid is positively related. So I came up with this idea that there's a phase shift. In fact, related to one of your countrymen, Ralph Martin's work.
Oh, yes.
Ralph Martin did the very first good oxidative stress study in Alzheimer's disease. He found there was an induction of glucose 6-phosphate dehydrogenase, a key enzyme of glucose utilization, but is important in generating antioxidant defenses. And some newer studies suggest that it actually plays a role in something like a warbird type effect. So that's new material. So back to this thing, I think that there's
quality research. Yeah. How do you
how do I generate quality research? Well, number one is you hope that your work is reproducible. If it isn't, that's square one.
That's number 2 and 3, I think, isn't it?
I consider that to be, and I'm not saying 100% reproducible either, but 95.
Okay.
Which is way higher than other people consider. It should be that anybody else could reproduce the work. Certainly you can reproduce it.
Yeah. Has anybody else replicated your recent studies, or previous studies? Has that happened, do you think?
I think so. There's only one study that I know of that people question, and it wasn't, and I didn't think it was a very important study. And then
That's good, isn't it? Isn't good to question other people's work?
yeah. No, it's
in
good.
a constructive way.
Yeah. But let me tell you, this is beyond stupid. Okay.
Okay.
Because I found that Lewy bodies in diffuse Lewy body disease have tau.
Yes.
And I didn't see them in subcortical ones. Okay. So I wrote this, one of the early papers. I remember I got the cases. I didn't even know how to identify these type cases. And I got them from Catherine Bergeron in Canada. And we did a whole bunch of antibodies, and it was kind of boring. And Pam Galloway was a fellow that worked with me. I said, why don't we emphasize the tau is different? So it was like a boring paper that we emphasized one aspect. Well, then they said they couldn't find it was tau. Well, number one, the positive result is always right. Okay. The negative result, I mean, that's why I meant it about stupid.
Yeah.
Later on, after people wrote that I was wrong, like in 2 or 3 papers, and my boss, Pierluigi Gambetti, said, oh, people are questioning your work. So I spent, on the original paper, I bet I spent no more than 2 or 3 days, because Pam did a lot of the work and I spent a small amount of time supervising her. Okay, new thing, I spent a whole summer redoing the work with more cases I got from other people. I got my own. I reproduced the work, and then I thought I need to have a collaborator that's more of an expert than me. So I collaborated with Licio Forno, Miss Lewy Body. And then what did she do? She published it without me. So I was really upset.
But the work was reproduced.
Yes, it was reproduced. And then it was reproduced by a lot of people. And they made a big story out of it, that it was actually important. I still think it's trivial, okay?
Okay.
That's probably the only study I know of that wasn't reproduced.
Fair enough.
And my PhD thesis, people have reproduced. In fact, I'm convinced they fabricated it to reproduce it, because they... well, I did some of the, not the naptoquinone, but the lipid peroxidation work I did in a very primitive way. It was all done by 10-layer chromatography and co-chromatography. And those identifications are not definitive. So what did they do? They published that they exactly determined the isomer I found. Well, I'm not even sure the isomer I found was right, because I didn't do mass spec or anything sophisticated. I just did standards. So that, I don't think I've ever been questioned on anything. When I do research, I do want people to be able to reproduce it and build up for it, but I usually try to do studies well enough that nobody would even think of it.
Okay. That's interesting. Yeah. Look, I'm just going to...
That's crazy. And that's intentional.
It is crazy. It is crazy. Well, it's all... I think that's a flaw in the system. I think all people should reproduce. It's a good place to start for a postgraduate program, is to reproduce this data and then move on.
I've received contracts to reproduce other people's data. When I was a postdoc, I received funding to reproduce a paper in Nature that was, I don't know if it was made up. And I found lots of work non-reproducible. Yeah. Lots. And this is Charles Piller has been writing articles for Science on this topic.
It's a serious issue in laboratories. I'm just going to move on.
You know why this is a problem?
No,
because there's no benefit of having reproducible data.
Oh, yeah,
none. It's zero.
It goes to the question about publishing, because publishing first is the most important. Publishing fourth or fifth is not. Is that
the best? Is to be first and best. It's actually usually hard to be first and best. The other way to do is to publish something very small to begin with and publish a more complete one after.
The field of Alzheimer's is moving with antibodies now, and there's meta studies showing that if you're on a blood pressure control, you've got a significant decrease in the rate of Alzheimer's. And what's your views of where the field is going to go to help people in the next 10 years?
I think within the next 5 to 10 years, for the most part, in terms of where the money is and where the people they're publishing, the beautiful people, okay, whatever that means, the people in the A group, and I don't consider myself to be part of the A group by any means, not even somewhere in the B- group. I think they're going to still be pursuing amyloid and tau because they're still they got enough traction with this vaccine approach. The antibody and vaccine approach they're getting approved.
Mm-hmm.
They're not killing a lot of people. They're killing some.
Yes.
And
it's expensive.
It's expensive, but the price will probably go down, and they'll probably figure out better ways to administer it. And I think that's where things are going to stay.
And what about blood pressure?
You're talking about the whole issue of lifestyle.
Well, I am.
The blood pressure. Yeah. It's all together. That's going on, and it's certainly getting more traction.
Do you think it's important?
I think the lifestyle piece ties in with what I was talking about, my work, because you're looking at the brain is one of the most energetic parts of the body, as is the heart, and they suffer from aging because they require so much of the body. And when we think about that, the brain has problems. Well, look at muscles. Muscles go downhill as you age also, no matter what you do about it. I mean, you may work out and stuff, but you're still going to end out with muscle loss. And the same thing, the brain, you're going to lose neurons. And the heart's function is going to go down. But all of those respond to nutrition, stress, good psychology, and blood pressure is a reflection of that. So these are all different aspects of the same thing. But, people don't usually want to hear that. They want something that's from a pill or, I'm going to flush this stuff out of my brain. I'm going to have a clean brain.
So exercise as well. People don't want to exercise.
Correct.
Is exercise a factor has that been shown for Alzheimer's to be part of the lifestyle components?
Yes, there are 4. You can. The complex version would be what Dale Bredesen does, which is to analyze 36 components. I don't think that most people are going to be capable of doing this. It's expensive, it's tedious, etc. And he claims he can reverse things.
Well,
it remains to be said. Okay, but the simplistic version is that you can distill it down to 4 things, 4 pillars. And I work with the foundation, the Alzheimer Research and Prevention Foundation, and you can have all of their documents, I think, for free or essentially free. Or you can go to Bredesen's and pay a lot and have a more complete thing. And it's exercise, nutrition, maybe supplements if you're not eating appropriately, having a reason to be alive, stress reduction. And that could be by many different things. I mean, he suggests meditation, and actually there's been intervention studies that have been funded. He funded, the director of this foundation funded the finger study.
What's the finger study?
It's the Finnish intervention study, of which it's prospectively modified people's lifestyle and found that it had a reduction in their dementia. More recently, he's doing a study with UCLA in elderly women, postmenopausal women, also intervening, lowering stress levels. lowering stress is also complicated. And my PhD mentor, the marine biologist, his daughter is an expert in this field, Alyssa Epple. And some stress is actually positive. And also, when you look at why certain diets benefit, it's also related to stress. The plant-based foods are not as benign as everybody says. They're full of toxins. And those toxins, they work to promote stress responses in your body that protect you. Same as dietary restriction and exercise. They're all stress, but you don't want to be too stressed.
No. Is that so that's two directions, the anti-amyloid and lifestyle. So that's and the lifestyle is just going to be hard to implement.
Hard to implement, you'd want to have it in a pill, and it's not going to be in a pill, although the elements might be. Because, that's, the old idea was that, these plants are full of antioxidants. But if you take antioxidant, you don't really have a major benefit unless you're deficient. If you're deficient, then they're necessary. But having a large quantity of antioxidants actually is detrimental because it dysregulates the homeostatic balance.
What about environmental influences? We're seeing now that there's forever chemicals in our system which seem to attack the immune system. I know this is new stuff and it's anecdotal, but it seems to be becoming more solid that the pollution in our environment is affecting our health.
The best data along that line is air pollution.
Yes.
Principally particulate air pollution.
PM 2.5s.
Yes.
I've seen that study of across America linking health data to Alzheimer's and Parkinson's.
Yes, Caleb Finch's work. And then the earlier work is Lillian Calderon in Mexico City. Studying health issues. And she studied mostly youth to study the brains of people that died of accidents that lived in Mexico City and found that they had lots of amyloid-like changes in the brain. And those findings have been reproduced in other locations throughout the world. Other types of intoxicants, I think they're much better for Parkinson's disease.
Yeah.
For Alzheimer's disease, the connection is pretty weak.
So when we're talking about pollution, and it's one of the things which isn't mentioned, it's more of a people are focusing on the structure of decreasing carbon rather than decreasing pollution.
Don't get me there. My wife says I'm too obsessed about it. Where I studied initially, my first lab that I worked in, I worked with the same person all the time, but we moved. Keeling, from the Keeling curve and carbon dioxide, was one floor above where I worked as a graduate student. And he'd always give a talk about global warming effects in his work when nobody cared. Okay. I'm not saying carbon isn't important, but there's so much more. I mean, habitat destruction, plastics in the ocean, which we don't even know what these microplastics really do to us. I doubt it's I don't think it's anything good. So I think we've concentrated too much on something. And the other part, if you're looking at climate always changes.
Yes.
Unfortunately, it's always changed. And the sun changes its radiation. The earth changes. I mean, if you look historically, and people die during these periods. It was not, right? Ice age.
Yeah. Yes, we've strayed a bit, but I think the PM 2.5s, the pollution, it's clearly it's
and
related
this is
to
controllable. These are more easily controlled.
Yes.
The modern society is dependent on huge amounts of energy. And at some point, I'm sure with time and research, we will find new solutions. But it's pretty hard to make up for coal and you don't know what in the effect of carbon dioxide. I mean, it's a nutrient for plants. Is it really changing? So I get all this stuff from Scripps Institution of Oceanography, where this stuff started, all the time. And of course, everybody's into the environment, and I get it. Is it good? No, because we'd like everything to be the way it is, but life never stays where it is.
No, it doesn't. How would you mentor, or what advice would you give to a new PhD student? Why
Biggest problem, when Charles Pillar called me about the issue of the amyloid star, and I said, well, I never paid much attention to it because I thought it was a bunch of crap. To begin with, it didn't make any sense, and I'm still not convinced that oligomers even exist in vivo. Okay, although I have papers that dealt with oligomers, I collaborated with somebody so I could add that as an element to my studies. And then he says, well, it was cited 3,000 times, so other people didn't ignore it. So then I had to think about what it was. So when you come to this, if people publish work that is a next step of what is expected or saves a field that's going down the tubes, which is what it was at that point. And people forget about it in a few years. It's very unusual that people revive back that there's a thing. So that's what I meant about reproducibility.
Yeah.
Doesn't end out having much benefit. It's more exciting to have that piece. So there isn't a reward. I've worked with Aaron Sorensen for years, a scientometric person, and our work together, which I'm not an author of, was published in JAD. It was a 2009 analysis of citation patterns in Alzheimer's disease. And he's worked with the NIH and other people about trying to figure out about how to improve quality or things. But I'm not so certain anybody's driving it anywhere.
No, they're not. Yeah. And I think what AI is going to may counter the good of old research and bad research dying, because AI is, you do an AI search and you get all these silly papers come up. I'll call them silly papers, and the A beta star comes up, and there's no, I suppose this is going to be difficult for people.
Yeah, and back to one thing, even though I spend a huge amount of time peer reviewing.
Yes.
I always, and a friend of mine that was a senior person, always told me that when I reviewed grants, because I was on a study section and I was chair of a study section, I said all the great things I was doing. And he said, you're not doing anything great. Is peer review really impart quality, or does it impart orthodoxy?
Oh, what's your view on that?
I think it's a mix.
Yeah, I think so too.
And orthodoxy is no different than, the problems that kept Copernicus or Galileo. And I wrote a small article about this, about why I got involved in a lot of the open access or borderline journals, is because I was really excited about them. I thought they were going to really bring about a renaissance of opening new ideas. And I consider science is part about rigorously testing things and documenting, but it's also about creativity of ideas that eventually become testable. And I thought that most ideas are wrong, okay?
Yeah, that's it. Hypotheses meant to be proved or disproved.
Yes, and most of the time they're wrong. But wrong does not mean that they weren't fruitful.
That's true. The examination of the idea is useful.
Exactly. Exactamento. So That is what I thought they would. They didn't do this, by the way, but I'm not certain if they weren't destroyed by the major journals at the same time. It wasn't a straightforward thing.
There's a trend now for many people to write reviews, which cite reviews, which don't cite original research, and so you're getting the opinion of an opinion. This original thought is being lost, I believe.
That I don't know. One of the things that's happened in my area is the consensus paper. So when you don't know what to do, you write a consensus paper and get 100 people as co-authors, and you have a paper that's very cited. I don't know. It does add to the field, but it seems to be kind of a dominant piece of what's happening right now. But I do think that orthodoxy is actually a big problem.
It always has been in science.
Yeah, it's not new.
So I'm going to wrap this up. Is there anything else that you'd like to impart to this journal? And it's our hope that one day that you'll join the editorial board of once we've proven ourselves. But yes, is there any last thoughts in this very wide-ranging interview?
I was having my physical 2 weeks ago, and my physician, I'm 71 years old, and he said, "Gee, are you thinking of retiring?" And I said, "I don't know." I said, "At some point, yeah, because I'm sure at some point I'll physically or mentally not be able to do my job." And he said, "That's really unusual." He said, "Almost everybody who comes in here hates their job." And I said, I like my job. Doesn't mean that I don't dislike various aspects of it. Science is a very rewarding field. It's just very rewarding because you're always looking for things that are new. And I think it's a lot of self-realization. And I don't know if that's so true with the other things that people have PhDs do would allow, because I've done them. I've done administrative and I enjoy doing it. What I said some of the time when I did this, I felt my brain was becoming like Swiss cheese, like spongiform change, because you feel like the things are kind of repetitive. Surprisingly, so far, I haven't found teaching undergraduates to be so repetitive. And I don't know why. Probably because they're just so energetic. It's probably based on the students rather than the content of the material.
Well, the students are very lucky to have you. And I wish more senior scientists, if I may call you a senior scientist, will engage with the student communities. I think that's the way forward. We need to impart all of the things you've said and questions. And I think I'll call the interview to an end now. And I thank you for your time and your effort and your passion towards science. Thank you very much.